My name is Goldman. I'm a first-year PhD student with Dr. Bill Plunge. And my talk today is on reducing waste evaluations in Cartesian genetic programming. So, first, so why should you care about anything I'm about to tell you? So why Cartesian genetic programming? I assume most people here know something about genetic programming, about how it does cool things about beating humans and doing some sorts of design, and is able to evolve complex things like programs and functions and stuff like that. Well, Cartesian genetic programming is a subset of that, and I'll explain exactly how it's different. That's able to outcompete tree-based GP, which is the normal one used by most everybody in the GP field. So why care about evaluations? If you ever have done anything with black box search, evaluations are king. Everything in the black box search world is on minimizing how many evaluations you take, because if you didn't care, you might as well just use exhaustive random search, and you'll get to the end eventually, and you win. So everything is about making the evaluation counts lower. And especially when you're dealing with real-world problems, that's really important, because sometimes it can take you hours or days to evaluate one individual. You're not going to get a whole lot of those done. So anyway, motivation. What is Cartesian genetic programming? So, it's graph-based. It uses directed acyclic graphs, which allows for this neat little property known as value reuse. So what does that mean? Let's use an example. So up here we've got this nice example of, let's say you have this function x plus y divided by x plus y plus z. That's what you're trying to evolve or represent in your representation. What you see on the screen is a standard Goza GP tree. And you might notice something a little bit redundant about this tree. Most notably, those two sections. The sections in the that are highlighted, they represent the exact same piece of genetic material. It's x plus y. The function needed it twice, but in order to evolve it, you had to evolve it separately in two places. It's harder to do this because, well, you had to evolve it twice. Now, if these were more complicated things, you would still have to evolve it twice. Again, it's harder to evolve. Also, it means that if you want to make minor changes that are affected globally, you'd have to change both simultaneously. You couldn't just change one. So anyway, that's where graphs come in. If you use graphs, it allows you to reuse the value that you computed earlier. So here we're showing, by making a graph, you get to condense the representation, smaller stuff, easier to evolve, all sorts of stuff. It's also more terse, all sorts of nice things. So anyway, how do you do the representation of these graphs? The genome is broken up into nodes, similar to codons, I guess, for the biologists. Basically, you have the nodes. Each node in the graph is represented by a collection of genes. So up here, you'll see there's zero index. So you have the 0 node, the 1 node, the 2 node, so on and so forth. So each node contains genes for what function it performs. Pretty obvious. You'll see function values and all those. So like plus, plus, divide, whatever. And it contains genes for where to get the input information it's processing. So for the first one up there, it's processing x plus, it's getting from the x variable and from the y variable. The next one is getting from node 0 and the z variable. And then the last one is node 0 and node 2. So this lets you get information from any previous nodes and then process them. You may have noticed there's an additional gene there on the end that wasn't part of any node. That specifies output location. So this is where the final output of this individual comes from. So there's this neat little property on these genomes, on the basically active versus inactive genes. Similar to how not everything in an actual genome actually is used, there can be large chunks of information that are completely unused by the genome. So for instance, this is our one from the last slide, which represents that original tree from the first two slides ago. You can have these extra little sections added in. I have changed absolutely nothing phenotypically, I guess, about this individual. I've just added these extra little chunks that are not ever read. And the reason they're not read is because the output location doesn't read from them, and any node that is read by the output location doesn't read from them, and on and on and on. So they're basically inactive. They're not used. Canonical GP or CGP uses a lot of these. On the order of 95% of the genome, they argue should be inactive. And what this allows for is genetic drift. Basically, it allows individuals to accumulate interesting things in those inactive portions save old modules in there, whatever. They claim it's good. Future work I have might say it's bad. But anyway, they're there. They're important to this talk. Evolutionary mechanism, the last little piece you need to know about how to get these things then to evolve, is CGP uses what I feel as though it's more of a stochastic hill climber than actual evolution, because it uses a population size of 1. So as an individual, it produces many offspring. It produces those offspring by doing probabilistic mutation, so each gene has a probability to be mutated each time an offspring is created. So for example, we could, at some mutation probability, apply, and oh look, those 2 genes got chosen for mutation. When you choose them, they have to change, so we make 2 changes. Also, you'll note that it's smart enough that when it makes a mutation, it's semantically correct, so all the connections, updated connections, functions are updated to functions, so forth. The way replacement works is it is on parent is replaced if it is the offspring replaces the parent if the offspring is no worse than its parent. And this allows for maximum amount of drift because as long as you're not getting worse, we want new over old. And so, again, there's lots of evidence that shows that's good for CGP. So in this case, because there's only modifications as inactive, you know these 2 have to have the same output values because we didn't change anything that could affect what they output. So you take the offspring. And what's interesting here, what I'm trying to identify here, is something odd. We didn't have to actually run these 2 individuals on data. We just know that they're going to have the same fitness. They're going to have the same outputs because nothing worthwhile changed. So, I consider these wasted evaluations. Why spend an hour calculating how good that individual is if I can, in 30 seconds, look at, or in split seconds for computers, determine that it was a complete waste of time to ever evaluate it. So in normal CGP, it evaluates all the offspring. It doesn't even care. It doesn't check. It doesn't even actually check if there's any genetic difference between the parent and the offspring. But what's odd is that the set of active genes is actually known. Some previous work has shown that you can get a significant improvement or a significant reduction in evaluation times if you determine what are active and only execute the active genes. Which again makes sense. If 95% of your genome is completely useless, why evaluate 95% of it? So, for those computer scientists in the room, it takes very little time, lower complexity, to do these checks. So it's not going to violate the bounds of number of evaluations counts more than anything else. And so as a simplification, I said just compare with your direct parent. Yes, technically you can have the same exact active genes as somebody who was 10 generations ago or one of your siblings, but the probability that's pretty low just from genetic drift and whatnot. So you're going to catch most of the cases by just looking at your parent, and it's also more efficient because then you don't have to store all the old stuff. It works out. So the nice thing about what I've formulated so far is you can actually get mathematical bounds on how often this waste would happen. The general idea you're trying to get from this is that the lines are different mutation rates, the X values are number of active genes, and the Y is the amount of percentage of waste. And you'll see some of those for those really low mutation rates, there's a whole lot of waste up there. That's almost 90% waste. That's horribly bad. That's a lot of waste. But that's just theory. You know, maybe in practice it won't come out that way. So anyway, as part of this research, I proposed 3 novel ways to prevent this waste, to avoid it, to get rid of it. So the first one is skip. It's completely straightforward. If you find one of these things, don't evaluate it. Just give it its parent fitness, move on. Simple, easy. So, again, using our original example, that was the original mutation we had. We look at these, we don't evaluate the second one, we just give it its fit, or we look at its active genes. As you can see, everything in the highlighted boxes are identical, so we just replace. Accumulate. If you find one that isn't actively different, mutate again. And so the idea here is that you just make these extensions of mutant, and it lets you accumulate mutations to these inactive genes. You're letting the inactive genes change faster than the active ones. So you do this, you check it, you apply mutation again, you get to this point, oh look, we now have some active genes that have mutated, we're done. As an added little aside to allow drift to still happen in this case, you look at these last two. The top one is actually identical to its parent, so we know it's fitness. The bottom one, we evaluate. If the bottom one's worse, we get rid of it, and we just keep the top one. This allows you to keep all that drifted, inactive change without, and allowing for neutral drift just the same as previously. The last case, by far, is different from the other two in that instead of looking for these wasted evaluations, it changes how mutation works to prevent them from ever happening. Basically, the idea is that you choose a gene at random. You say, is that an active gene? Nope. Keep going. Nope. There we go. We hit an active one. We mutate all three, or all the ones that we selected, guarantees that an active gene has to be changed, and now you have this mutant offspring who's guaranteed every time to be actively different from its parent, so it always should be evaluated. You evaluate it, you check it, you move on. So, I'm moving a lot faster than I thought. Is everybody, I'm not moving too fast, right? Nobody seems too lost? Okay. So, I tried this on some problems, and you'll notice that all four of these plots look really similar. So on the x-axis, you'll see mutation rate. On the y, the number of evaluations required to solve the problem, the median over 50 runs. And you'll note the blue line is normal CGP. And you'll notice it has this nice saddle shape every single time, basically the same shape, with the exception of the one in the top corner there. But basically what's happening here, it makes a lot of sense. So everything from, I'd say, from the saddle point to the right is when mutation rate is getting so high that it simply can't fine-tune its way to the answer. Makes sense. I mean, you know, if you're changing a whole bunch of stuff, it's really hard to optimize. Everything from the saddle point to the left is when it's getting so low that you're getting lots of wasted evaluations. And why do I know that those will be caused by wasted evaluations? Well, you look at skip and accumulate, which are very hard to see, I now admit. The green and red dotted lines that are basically along that other bottom dotted line. They basically hit that saddle, or they follow normal on the right side, but on the left, when mutation rate gets low, they basically just don't get any worse. They stay basically at the same level. And that's because they're not wasting evaluations. They're able to stay high quality even when mutation rate's too low. And then the last line that's completely straight across all the way is single, because I didn't mention it before, but single doesn't have a mutation rate. There's no configurable parameter. Oh, yes?
Sorry, is this showing number of evaluations in all these slots, right?
Yes.
Okay, so there's no comparison of fitness here?
All of these runs got to fully optimized on the problem we were doing. They're 4 binary circuit benchmarks, which is traditional for CGP. It was originally designed for circuits. But yeah, so the straight line all the way across is single. It doesn't have a mutation rate, so it gets the same results on all the mutation rates effectively because it doesn't use it. And so you'll notice that it's pretty well flat all the way across, or it's going to be flat, but now that it's actually better in a lot of cases. So some neat conclusions from this. So some neat conclusions. So skip and accumulate. They were better than normal. They were always better than normal. In all cases, they were at least as good as normal. That's because if the mutation rate was over a certain point, there were no wasted evaluations, so they could do exactly the same. And if there are wasted evaluations, they just do better every time. Yes?
How was the computational time?
So that is a little bit why I tried to sell you guys at the beginning about the evaluations being king. Because I still firmly believe evaluations are king. But for those people who are more interested in the computational time, skip and accumulate are faster than normal. Single is a lot faster than normal. Even when you count in the fact that it does less evaluations, because it doesn't have to generate as many random numbers, because it doesn't have to do as many operations, it actually ends up being a lot faster. But in all cases, all the techniques I proposed were both computationally faster and evaluations faster. Although they were obviously less, the computational improvements were less than the evaluation improvements because, well, there's overhead. You're calculating extra things. If I were to try it on harder problems, it would get more significant because sooner or later the evaluation is going to dominate everything else I'm doing. So like I said, skip and accumulate better on everything. The other nice thing is that they're actually less sensitive to the parameter, because I can just set it arbitrarily low and it will still do alright. And it's relatively similar to each other. From my explanation, it may not actually sound like these two algorithms should do the same. I mean, one is just skipping evaluations, while the other one is repeatedly performing mutation. They shouldn't be that similar, but the graphs beg to differ. And actually, well, it's a little involved. It actually ends up being that the two algorithms are almost identical. The difference is that in skip, you put off those future mutations to the next generation, whereas in accumulate, you're trying to do multiple generations worth of lineage in one individual, effectively. And so they end up working out to fairly similar. It's possible on harder problems I might notice differences. I don't think there will be terribly. So in general, skip is better than accumulate just because it's really simple. Alright, so single. Something I didn't mention on that last slide. So there's these 4 problems here. They go clockwise in difficulty. Top left is hard, bottom left, or sorry, top left is easy, bottom left is hard. The dotted line gets better and better. The flat line gets better and better as it goes around. So basically what I found is that on harder problems, it seems like single is doing better. This to some extent makes sense because single is better at doing really small changes to the individual. It's able to always make that one change, make small steps. And on easy problems, making small steps means it takes you longer to get to the answer, whereas on hard problems, it means you can actually get to the answer. So, that's cool that single is getting that improvement. It's also effective without tuning, which tuning is the bane of all EC. It is very expensive and a lot of times is actually more time consuming than just solving the problem straight out. So being able to do it without tuning, that's neat. And it's also less computationally expensive, as I was telling Professor Deb. It actually, from a computational standpoint, was easier, was less expensive than the others, than any of the other techniques by actually a significant amount. Some overall stuff. My results were relatively problem independent. Those 4 graphs pretty much look the same. So this implies that these results should hopefully be true on any problem you tried CGP on, hopefully. It matched almost identically with my theoretical expectations. For all these runs, I also predicted the expected amount of waste, and then I looked at the difference between skip and normal, and I saw that they were almost dead on the same. So it is doing theoretically exactly what I expected, so some more backup to my results, and then potential improvements by orders of magnitude. Those graphs I had were log-based. So those differences in lines were really big. So when the mutation rates got small there, it was by orders of tens to hundreds, sometimes thousands of times faster. Thousands of less evaluations when using skip or accumulate, or sometimes single. So lots of time for questions.
So, was your genetic algorithm using some form of elitism or something like that?
The only selective pressure, like I said, was the way the parents are replaced by the offspring replace the parents, and that is just every time a parent produces offspring, the best offspring is compared with the parent. If it is no worse, it replaces the parent.
These were lambda plus 4, right? Isn't that the way you ran it? Right?
Yeah. So you have one parent, it produces 4 offspring, takes the best of the 4, compare it with the parent, replace the parent if the offspring is at least as good. And that was the whole selection method, everything. So.
So that does enforce.
Yes. Oh, it enforces very strong elitism. But the way it gets around that otherwise nasty elitism is that due to the representation with all the inactive genes and all the drift and the way the mutation operator works, it's still able to do phenotypically large steps in what would be small space. So they've shown that as long as you allow for that drift, it doesn't seem to get stuck. It's still possible that on hard problems it will get stuck. I mean, that's always true, and I'm actually very suspicious of this 1 plus lambda I think the real key is that no one so far has come up with a good way to do crossover in CGP, and I think as soon as you do effective crossover, populations will again be useful, but until then, I think this is the best way to do it. Yes?
Are you worried about the fact that your single method in particular will never cross a fitness value?
So it still can, it's just less likely. And that is because of the use of inactive genes. So by significant difficulty, it is always possible that you could, in theory, let's say you have some individual, and it's using 50% active, 50% inactive. By very wide margins, it is possible to drift all of your genes to something new, then change your output location with one single mutation to that new, fully inactive individual, and get there. It's very unlikely, but it's always possible to make those steps. And so what I've found, at least, is that at least on these problems, it wasn't an issue. It is possible that there are problems where it will be an issue. I think that the way CGP works with this repeated use of inactive genes and its the effect that a single gene's mutation can have on the phenotype, it's unlikely that it will get stuck. I think that it's probably just as unlikely for normal to mutate the right 3 genes as it is for single to build up the drift and then get the right mutation. Yes?
Did any of the 4 problems you tested it on there, did they have the fitness value in them? Because I'm actually curious to see empirically how it would affect how quickly it reaches a solution.
It's difficult to say when you're evolving circuits what a fitness valley is. I'm almost positive they have plenty of them. Just because, I mean, for instance, the hardest problem is you take in 6 binary numbers, you multiply the first 3 by the last 3, and you get the result out. And your fitness is based on how many of all possible inputs, how many of them do you get right. And so it's very easy to construct something that is going to do most of it right. I mean, to some extent, something that always returns all zeros is going to be right a lot of the time. Mm-hmm. And so getting all zeros to even getting one of them right would probably be a fitness valley. Because it's going to be a very different shape from that to an actual multiplier. And there was also a parity problem in there, which has a similar issue. And, yeah. So I would be incredibly surprised if there aren't fitness values in these problems.
So do you have any feel for, if your process does 1,000 evaluations for what fraction of the time an inactive region becomes active in those evaluations?
What a wonderful question.
So I'm in the process, I'm actually working on a journal article right now that's an extension of this and work beyond this even, that is going to look at exactly that, because so the work after this is basically a claim that the reason it has so many inactive genes is not because it's necessary, but because the way CGP enforces its mutation, the only way it can have it is with that many. And then I tried to break that, and then the work that I'm working on currently is to look at how often inactive genes come back, and now that when they do come back, how have they been modified? Because I believe what's happening is it'll come up with something good, and then it'll come up with something good again, and it'll put the old thing into the inactive space, mess with it a little bit, and occasionally try to reconnect to it. And so it has all these different... No. And so I think it has all these inactive things built up that are similar to what it has in its active genes. It can make some modification and occasionally try to reconnect into them.
So, the next case that comes to mind is you're dealing with a single individual rather than a population.
Yep.
And I wonder if you use the same kind of approach in a population base, whether you would not see this 95% inactive stuff, but you would see that decrease dramatically because there are other ways of moving among alternative representations of
It is possible. The using 1 plus lambda is something that's been fought over, I guess, in the CGP literature. It was not originally that way. It's kind of from testing gotten there. And I think what it is that in the representation being used, the advantages you would get from having multiple individuals is offset by the fact that you want to really optimize that one individual. Like, that's what this does. It spends all this evaluations getting that one individual better and better and better. And if you had multiple individuals, you'd have to split that time across all of them. And the change in the selective pressure is just not enough to warrant the other versions, which is why I said...
I mean, yeah, it's interesting though, because I mean, we've seen, you know, in GA search, when you have a highly multimodal space, that sometimes it's worthwhile exploring multiple peaks simultaneously, and then, of course, there we have a crossover operator that can do those. Now, and with CGP, the difficulty, I mean, the reason why classical GP doesn't, you know, is able to use crossovers because they outlawed cycles, right? And so it's, well, actually even directed, they outlawed graphs. Other than just trees. So
so there have been some attempts at crossover in CGP, and in my opinion, they're all garbage.
Okay.
Well, one was just a one-point crossover to take those genomes I showed you and just break them in half and stick them together. And
that never worked.
That never worked. Never worked. There was another one that did something really odd where the way it decided each gene value was actually a floating point number, and then did arithmetic crossover on the air. It was garbage. I think that if you had something that was semantically aware, so you could actually take out the subfunctions or things like that, I think you could do a lot better, and I think you might actually be able to get back to having a population. But until somebody does something like that, which I guess I should at some point.
Think about that, because if you come up with an answer, I mean, we spend a lot of time in the early 2000s, you know, with function trees that we were evolving, and we needed cycles rarely, and couldn't do it well with you know, with a good crossover.
The sad thing about CGP is that it does allow value reuse, which with the directed acyclic, but it doesn't allow strict cycles. And so one thing that is missing from it is functional reuse. So like the example I like to use for over and over is if I need to square x, I can get a single subtree that squares x and then use its value all over the place. But if I need to square 6 different things, I have to have 6 different trees, all with the same shape but different inputs. And so CGP isn't able to do that. I'm hoping to actually, I'm probably going to move to push GP, which is able to do that.
Well, yeah, and classical GP,
can't do it.
I mean, well, COSA defined the ADF in order to be able to do that. Of course, the problem is that you've got to decide where you're going to have an ADF and what's going to constitute it. That's not an easy thing.
Nobody ever made it work.
In my opinion. It's not good.
Yeah.
So,
other questions? If anybody is curious, I have source code on GitHub. If you search the name of this talk, it'll come up on Google. And they should be able to redo everything.